Good morning, everyone. Welcome to our webinar with Copan on the topic the importance of sample collection in surface environmental monitoring. I'm Andrea Holzbeck, Supplier Relationship Manager at Avantor. And before we are moving to the session, I will let you know that the webinar will be recorded. The link will be shared with you after the webinar. If you have questions, please put them in the chat at any time. We will have a Q&A session after the presentation to answer your questions. If you have more questions after the webinar session itself, feel free to email at webinar@avantasciences.com. Our speaker today, Matteo, yes, maybe so the next slide. Yeah, our speaker today is Matteo Miriani. He's Senior Product and Marketing Manager for Copan in Italy. Nicolo cannot join, unfortunately, today. So, and with this, I hand over to you, Matteo, to start the presentation.
Thank you so much, Andrea. Before starting the presentation, I would like to thank you all for taking this hour to listen to today's webinar on surface environmental sampling. Obviously, I would like to thank a lot also our colleagues from Avantor for giving us as a Copan the chance to present today our solution for this specific application. As a first slide of today's meeting, I would like to start with a definition. So the definition of the environment, which is basically looking in the vocabulary, defined as a surrounding or condition in which a person, animal, or plant lives and operates. Clearly, the environment is composed by different elements, so person or animals, plants that lives in that space, the air, which is filling actually the space, as well as the surfaces, in which all these things are in contact. Clearly, there is a continuous exchange of material between all these three main points, these main contributors in the environment. So if there is an exchange of material, this exchange of material can be beneficial in certain cases, but also in other cases should be avoided or controlled in order to avoid the spread of one thing that came with a person that should go to the surface and then to the air and then back to person. So that's the basis of, and I think that I should not have to explain too much to you today on environmental monitoring. And that's very important because if in that environment I need to manufacturing a product or I need to keep a person in a special condition, a special clinical condition, if there is something harmful that is bring to the space by one of these three elements can be spread to the products or to the person. And in some cases, we need to avoid or we should monitor this process. So basically, going on more on the specific on surfaces, which is actually the topics of today's webinar. It's a common knowledge that work surfaces can become contaminated with chemicals, biological or radiological agents in different circumstances. And this could be just an emergency work activity, it could be an accidental contamination, an intentional contamination, or in most of the cases, just happens during the natural occurring process on that specific process. And again, if the agent that is that contaminates the environment, the surface, is a harmful substance, and the definition of harmful can change depending on where you are actually acting on the specific field where you need to monitor, the outcome can be extremely dangerous or at least threatening for the general healthcare condition. Because if we think about clinical settings, the outcome could be, for example, which is something extremely concerning in the last year, the spreading of multi-resistant drug microorganism, which in a clinical settings can be present, can be brought by a carrier person, colonize surfaces, and then contaminate and infect a person that should not be infected, especially in a certain clinical or physiological condition. That's just an example of the clinical setting, but we can go through many different type of thread, like in the food industry. Because if, for example, poisoning bacteria or harmful bacteria, pathogen is present on the surface, can spread and contaminate the whole production of the day or the batch. And this can lead to food poisoning or of the person that they are going to eat that, or food recall with extremely high cost in terms of management of this food recall. Just the last example where the environmental monitoring is extremely important is the pharma industry, where if there is, for instance, a contamination of a product with a certain microorganism or contaminants in general, this may have a huge health threat on the person that they are going to take that specific drug, as well as a drug recall, which again, it's terrific and dramatic cost for pharma industry that they are going to do that, as well as image damage. Clearly, that's a big problem, especially for pharma industry, because if we look to the data from FDA, US FDA, microbiological contamination was the leading cause of recall during 2022. So again, it's an extremely and a paramount point, the monitoring of surfaces together with air and person. But again, for the scope of this webinar, we are going to be more in detail or more in the scope of surface analysis. The problem is that surface monitoring is something extremely complicated. Because surfaces are not actually standard, you cannot standardize a surface because they may change in terms of material, they may change in terms of dimension, the accessibility of a surface that need to be monitored and sampled could be very poor, and normally the area less accessible are normally the ones that are more at risk to generate contamination, especially microbiological contamination. The surface properties is another very important point, because clearly smooth surfaces are easier to clean, and so they are less, so to speak, at risk to generate contamination compared to rough surfaces, which in some cases for production needs cannot be substituted by other. The surface status, dry surfaces are less prone to generate microbiological growth and contamination compared to wet. And last but not the least, on surfaces may be present substances that can interfere with the analytical method. And for the specific scope of microbiological surface monitoring, these interfering substances are exactly the same one that are normally used during the cleaning process of the surfaces. Because if traces may remain on the surface during the collection, then these traces can reactivate, so to speak, and going to reduce the viability of bacteria that needs to be cultured, going to affect negatively, with false result, the final outcome. The problem is that how we should or how it should be the perfect device for sampling surfaces. I want to first of all say something or at least doing a disclaimer. The perfect collection device doesn't exist. So all the technology that or all the way that we can use for sample surfaces shows some pros and some cons. But generally, from a very generic perspective, the major feature that a good or perfect device for sampling fast surface should have is that this device, this methodology, should recover as much as possible sample, because I need to understand exactly if there is something, even in a trace, I have to be able to recover as much as possible these contaminants. The collection device should also be able to neutralize eventual interfering substances, the one that we were talking before, because if I'm collecting something together with my analyte, with my microbiological analyte that can interfere on its detection, that's a big problem. So that's why I should be able to neutralize or remove in some way the interfering substances that can affect negatively with the analytical process. Very important, the sample should be released by the collection device in a quantitative way, because I should not also, I have to be also able to not only detect in a qualitative way my analyte. So for me, not necessarily the information yes or no is sufficient, but I should be in a situation that I need to count the amount of bacteria, for example, that I collected for a specific surface or a specific dimension of surface. Very important, I have to be, this device should be able to reach any area that I need to sample. And as we were talking before, the poor accessible area are normally the more at risk to generate a contamination, because if it's complicated to access to an area for taking a sample from that, it's also complicated on the other side the cleaning process of that specific area. I should be able to preserve the viability of the microorganism if we talk specifically for microbiological quality assurance for the longest time possible. So first of all, I have to be able, this device should be able to preserve the viability, full stop. And also, it's very nice to have to preserve the viability for the longest time, because not necessarily we are in a situation to proceed to the streaking of the sample on the plate or into the broth, enriched broth, just after the collection. But I may be in the situation to wait for many samples during the sampling activity during the day in order to have a batch, or I should be in a situation that if I don't have the facility, internal facility to analyze the sample, to ship this sample in an external lab. In this case, it's becoming fundamental the possibility to preserve the viability. Because if what I collect arrive that to the laboratory, again, that specific sampling step was completely useless. And last but not the least, clearly it should be easy to use, because in many cases I'm not in a comfortable situation during the sampling step, because if I'm in a sterile area, I will be dressed in a way that also the step of collecting a sample, it's not the easiest things in the world. So easiest is the way to use of the sampling device. Better is the process, and less are the risk to having a spillage of liquid, for example, or to jeopardize the sample collection itself. So, what are the main the main way or the main technology to collect a sample from a surface, especially from a microbiological point of view? So we have basically three main ways: so contact plates, wipes and sponge, or swab. As already said before, there is not the perfect collection device, because every collection device show pros and show cons. In the case of contact plates, they are, without any doubts, very easy to use. They show, generally speaking, very good recovery, and they don't need any kind of additional sample management, because they have just to be incubated. The problem of the use of contact plates is the fact that they can be used only for flat surfaces. They contains nutritive media, so the cleaning of the area after the sampling should be adequate to enough to remove any material that then pass on the surface that can generate themselves a contamination or a growing substrate. Can be used only for bacteria, so only for microbiological collection, clearly, and potentially you may need multiple plate depending on the quality assurance that you need to do. You may need multiple plate for doing multiple collection on the same surface. Moving to wipe or sponge, they are definitely probably the biggest, the nicest way to collect a sample from big surface. You can use, without any doubt, for collecting a sample from irregular surfaces. The problem is that they are honestly quite complicated to manage. Because they are quite prone to get contaminated, because you need to physically touch. But also the problem, they show some problem in the analytical processing of a sample collected with a wipe or a sponge, because they are generally not quantitative, or very poorly quantitative, and they are also complicated to analyze. The last approach that you can use are swab. In this case, I will start with the cons, which is basically the fact that with the swab, the sampling area that from where you can take a sample from generally is not bigger than 100 centimeters square, so a square of 10 per 10. So they are not ideal for big surfaces. On the other hand, swab are perfect for irregular surfaces, are perfect for small hole, if you need to enter into a pipe, for example. They are quantitative, so you know exactly the release capacity of the swab, so you can very easily quantify the amount of sample that you eventually collected from a surface. They show normally a perfect recovery, and you have the capability to choose to elute the swab in some kind of medium, and choosing the volume of this medium. And this gives you the possibility to be extremely flexible on the analytical process that you want to do. Last but not the least, they normally are designed for microbiological collection, but not only, because from swab you can basically collect virtually any kind of contaminants that you need and you want to take the sample to monitor. So, coming to Copan solution, as a company, as a market leader in collection and transport device for microbiology, we developed, and we are very proud to have Avantor as a partner for the commercial part of this product. We developed the SRK line, our surface sample collection and transport solution. Copan SRK is a line, it's a product line, and if you are interested, I invite you to visit on the Avantor page, the SRK specific page, which include a comprehensive, tailor-made and ready-to-use collection and transport device. They are developed for and designed for specifically environmental microbiological contamination in a broad range of environment, such as food production, pharmaceutical site, and also in the clinical space, so in the hospital wards. SRK line is a range of 30 products to meet all the different needs of surface collection, because as we saw before, surface cannot be standardized, so it's really complicated to have one device that does all. So that's why we develop such broad range of product in order to allows any user, any potential user, to find out the right product for their specific needs. And the SRK were developed and manufactured in compliance with different guidelines and regulation in order to fulfill also the regulatory part in a easier and faster way. So to basically give you an overview on the SRK line is basically composed by a collection device, which the top level products in the line are based on the proprietary technology from Copan, which is called flock technology, that we are going to see in the next slides, and a medium, a neutralizing medium called SRK, which may be used as a pre-wetting solution for increase the collection capacity of the swab on dry surfaces, as well as for the transport of the sample after the collection. And if we want to go a little bit more into the collection part, so again, two main step. I need to collect and I need to transport my sample. So, and as said before, the collection part, the top level products in the lines are all based on the Copan flock technology. What is the Copan flock technology? The Copan flock technology is a proprietary way technology to manufacturing a swab. This technology basically allows to spray thousand and thousand of nylon microfiber on a stick, creating a swab which is characterized by this very hairy structure, which magnify and amplify the surface area where the sample can be collected. So it has a huge impact on the absorption capacity of the swab itself compared to traditional swab, but also mostly on the release capacity. And that's why it makes this device a very a real quantitative device that collect a certain amount and release most of them. We are talking about, depending on the situation, but we are talking in the range of 90% of what is collected is actually released into the medium or wherever the swab is then used for the analysis. And that's a very good, a very important point. Also, the flux technology allows to do the way how is manufacturing to exploit the capillary action between the different fibers. And this is very important because all the volume of the swab is can be used for collecting the sample and not just the area that is physically touch the surface, because the point that is actually of the swab, which is touching the surface, then absorb and move all the sample throughout the whole volume. So again, you can increase dramatically the absorption capacity. Last but not the least, very important, is made by nylon. So it's made by an artificial, a synthetic fiber. And this is very important, especially if we talk about microbiology and bacteria viability, because doesn't contain any kind of substances into it that can have inhibitory effects like the one that are normally present in cotton swab, because cotton swab are normally bleached during the production for making them white, and these chloride substances may remain, or traces of chloride substances may remain on the swab, and that's have actually a dendrimatic effect on the viability of the sample. This is a very interesting and very nice picture that I took from a very nice paper published by Alexander Probst in 2010. And on the left, you can see the microphotography of a flux swab. Compare on the right the microphotography of a cotton swab. As you can see, the structure is completely different. You have this very hairy structure on the left on the flux swab, while it's a very close and compact structure on the cotton swab on the right. And this increases the capacity, the collection capacity, because the surface area is dramatically higher for the flux swab, while it's very small for the standard swab. And last but not the least, also the medium that can be used for releasing the material from the swab is able to enter in between the fibers and allowing them to be released from the swab itself. And clearly, there is also a huge effect on the performances from the same paper. The two black and gray bars are the recovery efficiencies on, in this case, on spore, Bacillus spore. And this was compared to standard cotton swab, which are the white bar. And you can see the amount of sample that can be recovered is at least twice compared to the standard swab. So again, the capacity to collect a sample and then to release for their detection is dramatically higher for flux swab. So we, as a Copan, we are proud to have this technology in house. We actually, it's our patent, and the application of this technology for collecting a sample from a surface really can increase the quality and the amount of sample that then can be detected. But clearly, collecting a sample is not enough for assuring best result, the best result that actually you deserve. But you need actually also to have a very good way or very good medium to pre-wet the swab for collecting the sample, for increase the collection capacity from a surface, as well as for transport the sample that you collected from the surface itself. And our solution is to couple again the flock swab together with the SRK solution. The SRK solution is actually an isotonic saline solution, which is able to inactivate sanitizing agent residues that may be present on surfaces at the time of collection. So you are removing all that inhibitory chemicals that can kill the bacteria, so making them not detectable anymore by culture, as well as allows to preserve the viability during the collection in the period between the collection and the analysis that not necessarily are immediate. SRK solution, as said before, is isotonic minimum non-nutritive medium and contains lecithin and polysorbate 80 for the inactivation of chemicals. And in the table at the bottom of the slide, you can see the different type of chemicals that we tested and also the concentration that the device is able to neutralize them, but also contains molecules like the sodium thiosulfate and sodium thiglycolate, which is able to preserve not only the aerobic flora, but also the anaerobic flora that can be extremely dangerous in certain circumstances, like the pharma production or the clinical environmental testing. SRK solution can be clearly is designed for preserving the viability, but the sample collected in SRK solution can be analyzed also using molecular assays without any problem. Again, SRKs allows you for not rushing to streak the sample on the plate, but you can transport actually the sample at room temperature between 2 to 25 degrees. So you don't even need a cold transport or a cold chain. And you have 24 hours of time from the collection to the time where the sample needs to be stricken on a plate. And also the data that we have are on a multiple, on a very broad portfolio of microorganisms that are normal or pathogens that may be present on a surface that I want to detect, to detect. Okay, SRK configuration. SRK configuration, I love to define the SRK line, or to explain the SRK line as a Lego, because are basically a mix and match of different building blocks. So you can choose your final product, or you can, in other way, you can build your own product, taking different bricks of different color or different shape, depending on what you need to do to create or to find the right products that fits with your needs. And let's have a look what are actually these different building blocks or bricks or Lego bricks for making your final products. So you can basically choose the applicator type, so the swab type, and material as well. So in the line are present products with standard flock swab, the one that we saw before. We have products that includes flock swab with pre-moistening sponge. So what is this? It's basically a sponge. It's a product, and the flock swab is put into a tube. It's a soft tube, and at the bottom of the tube you have a sponge which is imbibed with the SRK solution. So for let the for pre-wetting the swab, you don't need actually to physically dip the swab into the SRK solution, but you have just simply to push the bottom of the tube. The sponge will release the SLK solution that then goes to pre-wet the sample, the swab, and then at that point you can remove the swab from its tube, and you already have a pre-wetted swab for taking the sample from the surface. And for example, this is extremely interesting because compared to other product present on the market, you have a lot of liquid, so you are always sure that they are not going to be dry. So you are always sure about this. That's the swab can be always at the best situation in terms of humidity. And for example, if you are working in a sterile area, it's this process to pre-wetting the sample can be done very easily with gloves or the wearings that are normally used in a sterile area. So to wrap up, we have flock swab with a pre-mostering sponge to facilitate the pre-wetting step, but also we have fiber swab, standard fiber swab, in case you may prefer this type of swab. It's made by viscose, so again, it's not cotton that contains chloride substances or harmful products for bacteria. It's a pure product, but in a standard technology manufacturing way. Last but not the least, we can provide a sponge spatula for larger areas. Then, once you choose the best collection device, the best device to collect the sample, you can choose the transport medium. Clearly, the SRK medium, the one that we saw before, it's our all-rounder medium, collection medium and transport medium, but also we have in our portfolio lit and brought. For example, if you need or if you work in the cosmetic analysis, in cosmetic industry or wherever, lit and brought is specifically asked by testing guidelines. So we choose the collection device, we choose the SRK, the transport medium. You can also eventually add a third building blocks, a third bricks to your final products. So an enrichment medium. In the portfolio, our SRK portfolio, we are available TSB for generic enrichment broth in the case that you want to run a sterility test. We have a Hal-Fraser broth for Listeria detection. We have a Rappaport Bacillus broth for Salmonella detection, as well as a buffered peptone water for a generic enrichment like for coliform. In this case, you can remove the tube with the SRK and add the enrichment broth. So you can do your collection and directly snap the swab into the enrichment broth, skipping basically the transport need. Last but not the least, we also have a product with a special feature like the triple packaging for which is mandatory for pharma industry, especially for sterile area sample collection. And the all triple packaging in the Copan products are VHP certified, as well as we have sterile control dimension frame, 10 per 10 or 5 per 4 centimeters, in order to collect a sample from a specific or controlled dimension surface. So how is it fit the SLK solution in the perfect device for sample surfaces that we saw before at the beginning of the presentation? Should it recover as much as possible sample? Yes, it does. In this graph that was taken from a paper published on the Italian Journal of Food Science. Basically, there was a comparison done on with traditional swab and flock swab in recovering E. coli from plastics and steel surface. And in every cases with different bacterial load, flock swab always overperform compared to traditional swab. So flock swab that you can choose in the SRK line always allows you to recover more samples. So if you have a strict parameter to respect, flock swab allows this. It neutralizes eventual interfering substances. We saw that SRK contains molecules that are able to inactivate and to neutralize different antimicrobial family compounds like oxidating compounds, quaternary ammonium, biguanidine alcohols, and phenolic compounds. And these allow you basically to be sure that if any substance or any interfering substances are present, these are completely removed from the sample or neutralized from the sample. Also, the flock swab is proven actually to release the sample in a quantitative way. So again, on the same paper from Italian Journal of Food Safety published in 2016, the authors were able to show and to prove that you have, in most of the case, almost a complete release of the sample from the swab itself. So you have basically what you collect, what is present on the surface is collected and then is released into the final medium. With swab, clearly, and with the flexibility and the resistance of the flock technology and the swab from Copan, you are basically able to reach area that in a very easy way that needs to be sampled. So you won't be any more constrained to take sample also from a very complicated area. We have users that are happily using our SRK product, for example, for the testing of endoscope for checking and they are taking sample from inside the endoscope for checking the cleaning procedure of them. Again, the SRK allows you to preserve the viability of the microorganisms for a long time. You can transport at room temperature up to 24 hours, and that's formally validated. So this is not an off-label, it's formally validated from us, so we are taking the responsibility about that. And this basically allows the safe transport from the collection site to a service lab, if this is the case, or allows a better lab organization because you can do a safe batching procedure, because you can batch all the sample in once, and you can then work all the sample in a more straightforward way. And allows the retest of the same sample, because if for any case something goes wrong on the first trial, you can retest from the same sample. And it's easy to use because, for example, for what we saw before, the pre-wetting tube that we have, the one that you are seeing into the picture, allows you to be extremely efficient, or it's extremely easy to use, even with gloves or strong dressing specific or of certain needs of collection needs or collection area. So to conclude this presentation, surface sample collection is a very challenging, because the variability of the surface properties and of the microorganisms that may be present are really not allowing very high standardization, does not allow actually a good standardization of the collection process. It makes them quite difficult. Different methodology for sample collection are available, of course, all with pros and cons. So it doesn't exist the perfect collection device, but definitely swabs, despite showing certain limitation, for example, on a big area, are definitely one of the most universal, because allow you to collect different type of analyte, microorganism, and more. And in case of microorganism, you can analyze by different technology, culture, PCR, or whatever, and also allows you choosing the right volume of solution also to apply different culture technology. Clearly, not all the swab are equal. There are swab that performs better and swab that perform less well. It's important, very important, to know actually the performance of what I'm using. So, and once you have the knowledge of that, it's important also to understand if it's exactly what I need for collecting and monitoring the surface that I need to collect the sample from. And finally, Copan SRK bundle the high collection performances of the flock swab with the neutralization and transport capability of the SRK medium. And this allows to improve and standardize most of the surface collection needs in any sector. And with this, I would like to really thank you all for your attention. Please do not hesitate to contact the Avantor colleagues and to visit the webpage of SRK on Avantor webpage. And obviously, we are available for any kind of question, if any, after this. Andrea, I don't know if you want maybe to conclude with these two slides, and then we can open the Q&A session.
Yes, maybe you would first open the Q&A session, and then I do the conclusion after the webinar itself.
Of course, absolutely. So I see seems 6 questions. So the first one, where we can find the data sheet and certificate of these products. These are available throughout your contact point in Avantor. And we can provide basically all the all the certificate that we have. What is the shelf life or this product? It's depending on the product. It range between 15 to 36 month. How is detection of microbiological contamination in grade A? With flux swab compared to contact plates. That's comparable, and depending clearly on the type of material, the surface that you need to take the sample from, which in most of the cases here is steel. It may be also it's not inferior, and in some cases superior. Clearly, it really depends on case by case, also on the type of the contact plates that you are actually using. But we can follow up with data on this, because we have in-house data, and we can provide. You use swab for detection of pathogen, but with a swab, isn't possible to analyze a big area. That's in fact one of the limitation of swab. But actually, this can be overcome by taking different swab that can be all pooled in one transport tube, because the biggest problem is that over certain dimensions, swab tends not to absorb anymore. But you can use more swab and pooling in one transport tube. So that's very true, but there are ways to overcome this. Also, clearly, this is the reply to this question will need a very long discussion, but also, for example, there are work done on multi-drug resistant organisms in the clinical space, where basically you can find representative area from where you can actually get a good idea on the real contamination status of the surface. But yes, that's for sure one of the problem. Last question. Are the media use control like described in ISO 11133? I will follow up with this question in a second stage. I need to double check with my technical guys, and I will follow up with this after the call. There is another one that just came. So the swab that you propose for a user who is collecting a sample and then taking better result, you can also detect food pathogen. You can detect food pathogen with either flux swab, but also you can use a standard swab again using the same SLK solution. So there are reference code that includes a flux swab, that are reference codes that includes a standard swab, and it's really up to your needs to decide what is better for your needs. But you can use either. From our experience, flux swab are I would say more used in the pharma, okay, while standard swab are more appreciated by food industry, But again, it's really it's not that's a commonality, I would say, but it's not a rule. So we do have actually very happy food industries that are using SRK with flux swap. Will the slide be shared? Yeah, we do have. You will have the recorded presentation available. Okay. I don't see any more question popping up.
Okay,
Okay,
thank
I
you
will
very much,
rush.
Michael.
Thanks to you and thanks to all the attendees, clearly. And Andrea,
Yes,
it's yours.
thanks a lot for the presentation and thank you all for listening. I hope you had a very interesting session. You will receive an email now after the webinar with a link to the recording so you and your colleagues can listen again whenever you want. And you find the recording also on our website under events in the webinar corner. Also, a survey will come up to you after the webinar. Please share your thoughts there. Every attendee will also get a certificate of attendance afterwards. And last but not least, if you want to contact us, please send a message to webinar@avantosciences.com. So with having said this, thanks all, and I wish you a very nice rest of the day. Bye.
Thank you.